Arguably, there are no cooler cells in your body than the neuron. Neurons are fascinating things to think about and consider, and what I'm going to do in this video is go over the introduction to the anatomy of a neuron and also describe how neurons work in practice. And what I have here is a blank, somewhat labeled diagram of a neuron, and this is a very simplified version because we can clearly distinguish between the parts of it, but in practice, if you were to look at a brain, for instance, you would find neurons that are just clustered together in huge messes of spaghetti, and sometimes figuring out what the different parts are is a lot more tricky. But for the sake of introducing the concept, this is what we're going to go with. And so the first thing you're going to note about a neuron is that it has a nucleus, like any other cell you have, and it has a cell body that people refer to as the soma. And where things begin to get more interesting is we now see that this neuron has this kind of long branch here, which we refer to as an axon. And on these axons, we find stuff that's generally white, and it's wrapped in something referred to as myelin. And the purpose of this myelin is to act as insulation. And myelin is composed of types of cells. So we're not just looking at one neuron here, we're looking at multiple cells. The myelin is made of Schwann cells. And Schwann cells are a type of glia cells, which is a generic category of cells that their purpose is to support neurons. And I like using the reference that neurons are a lot like Hollywood celebrities. Neurons are the whole reason we can think and even listen and watch this video, but a neuron by itself is pretty non-functional, so we need support for them. We need glia cells to help our neurons out when they need it. And so what we're going to note also about these glia cells is that they don't have complete coverage of the axon, and because of that we see these little nodes or empty spaces, and they're called nodes of Ranvier. And nodes of Ranvier are where ions will flow in and out of the axon. They'll enter and leave the neuron itself. Now, to take another step back and think more about what is going on in neurons, we know that neurons are communicating between each other somehow. And therefore, there must be some kind of sending part of a neuron and a receiving part of the neuron. And so if we go to the left here and we look at the far end, what we find are these things called dendrites. And what dendrites are just receivers. And so dendrites will receive stimulus from other neurons, and the other neurons would be referred to as presynaptic neurons, and they will interpret the electrical or the chemical signal that has been received, and they will do something accordingly if a threshold is reached, and I'll get to that threshold later on. But let's assume the threshold was reached. What would happen next is the signal would travel through the dendrites, through the soma, and down the axon, and we would see a consecutive firing of, or a wave of ions flooding into the neuron along the axon in this direction from left to right. And once it reaches the end of the neuron, and this is referred to as the axon terminal, there's going to be dendrites to other neurons. And those other neurons would be called the postsynaptic neuron. So these are relative terms. So I can get more into detail with this, but we'll just call this dendrites of neighboring neurons. And so the signal would continue to pass through the network that's been established of neurons. Now, I'd like to talk a bit about the action potentials that are occurring in these neurons, because it does help solidify the concepts that are going on here. So if we were to look at the junction between an axon terminal and a dendrite, that is referred to as a synapse. And the space between them is referred to as the synaptic cleft. And the synaptic cleft is where the electrical signals or the chemical signals, such as neurotransmitters like dopamine, will actually be transferred. And so going more into what is happening within that synaptic cleft, if we were to take a look at the action potential, and we were to plot the relative voltage, and we know voltage is a potential difference between the nodes of Ranvier. So if we were to look at this region here, for instance, and we wanted to look at how the voltage, if we had a multimeter plugged between the inside of the neuron and just outside of the axon, it has a resting potential of approximately minus 70 millivolts. And what that tells us is that this minus 70 indicates that you have more positive ions outside than inside. And establishing this electrical gradient comes at a cost, and that cost is ATP. And so what you'll find within the axons of neurons are things referred to as Na plus and K plus ATPase pumps. And intuitively, you should see this and think that you're pumping neurons where they, or I'm sorry, you're pumping ions where they don't want to go, and it comes at the cost of ATP. And so what the cell does is it couples a spontaneous process, which is the conversion of ATP into ADP, along with the pumping movement of moving a sodium ion, for instance. And so with these sodium ions, we're coupling an ATP reaction to it, which allows us to push the Na plus ions out of the cell, which establishes this negative relative potential difference inside of the neuron. And there's another key figure to remember here, which is minus 50 millivolts, and this is what's called the threshold. And so this threshold is the potential at which the dendrites will actually respond. And so if we look at this and we had one dendrite fire, we might increase slightly up to, let's call this minus 60 millivolts. But if nothing happens, the ATPase pumps would keep pumping, and the potential difference would fall back down to minus 70. But if we were able to get just to 50, then what would happen next is we would have a huge rise in the potential difference. In other words, an influx of Na plus, and there's also calcium ions present here, rushing into the neuron itself in these, around these nodes of Ranvier, and we would see a skyrocketing or a very positive increase of the potential difference. And then what happens at the top is we now have potassium gates open. And the purpose of having the potassium gates open in these neurons is to allow us to begin to come back down to this kind of minus 70. Resting potential. And so if you didn't have these K plus gates open, the whole point is that these Na plus and K plus ATPase pumps would have to work a lot harder than they do. And then that is what happens at one node. So if this happened at T1, what we would find in the neighboring node after the action potential is reached is that then it would fire. So after the action potential rose, you would get the next neuron firing. And just to wrap things up, because you can get very deep into neuroscience and it's very cool stuff, I think this is probably a good place to end things. And I just want to emphasize here why we want these nodes of Ranvier in the first place. And the whole point of that is that we can see visually how much less area we need to establish this minus 70 millivolt potential difference between. And so if we didn't have the Schwann cells providing insulation, it would require even more energy to establish these kinds of electrical potential gradients, and your brain would need exponentially more energy to function, more ATP to pump out the sodium ions. And so I hope you guys find this video useful. Let me know if you have any questions, and thanks for watching.